we're going to talk about the biorepository now. And this is, like I said earlier, why when you get 12 tubes of blood, maybe 8 of them are going to this group here. So we're going to talk a little about what they do and how they keep track of them. We're going to hear from Dr. Laura Rassenti, who points out that she was born in Florence, Italy, so that must be important to earning her PhD in molecular genetics from the University of Arizona. Molecular aspect of a wide career. She dealt with multiple sclerosis as a postdoc at Stanford, and she's been working in the lab with Dr. Kipps, and she corrected it. It said 25 years, she said no, 30 years. She's the director of the TissueCore for the CLL Research Consortium. Donna Neuberg has her doctorate degree in science. She's a senior statistician. So she's going to tell you how important that is for leukemia, lymphoma, and also for benign hematology on the other coast in Boston at Dana-Farber Cancer Institute. And she's led the CRC biostats course since it was formed. And Andrew Greaves, who you've all met, says I should introduce him as a MacGyver in training. So Andrew, besides taking care of the audio-visual, moving the chairs around, arranging a vegan lunch for me, is also, his real job is, and I'm going to get this wrong, is a biomedical statistician. Biomedical informatics. So he puts everything, I told you I get it wrong. So he puts everything together in terms of making sense and being able to pull the data. So I'm going to let these, they said to introduce them as the Three Stooges. TissueCore. And so,
thank you very much for that introduction, and I really want to thank everybody for inviting me to speak at this very, very important and worthwhile event. And it's my honor to tell you what we do with your blood and go through the process of how we accumulate the data and the information that helps us make discoveries in CLL. And then Donna will talk after me, and then Andrew will show a movie of exactly motion of how we do what we do. So I just want to give you a kind of a listing of what are the roles of the biorepositories. First of all, it provides a national repository for samples that are received from CLL patients. It receives, processes, and stores blood samples during the disease course of these patients in a uniform manner. We characterize these samples according to standardized operating procedures. So all the samples, all the procedures are done the same way with all the samples in the same lab. And we also store the corresponding clinical data, including clinical data from CRC trials that is associated with each of these samples, the disease course of the patients. And that's where Andrew comes in with the relational database for the CRC. And a more important project, what we do is we redistribute these CLL samples to specific CRC investigators for specific studies that expedite the discoveries and novel therapy for this CLL. Now, I kind of want to look at this like a stocking on a pyramid. First of all, on the left side you see the time, and this has been going on for almost 14 years, if not more. And at the beginning, we get samples from all of it. We get the blood plasma, we get the bone marrow samples, we get samples from longitudinal studies. So that's the very first step that occurs. Then above these samples, with these samples, we make DNA, RNA, and then we determine, as you know, the VH mutation status. We do whole genome sequencing, so we do the genetics on these samples. Now another ladder on these samples, what we do is we determine the surface and intracellular marker expressions by flow cytometry. We look at the percent B cells, percent T cells, ZAP70, CD38, CXCR4, or many of all those molecules that Dr. Kipps described this morning that allows the B cell to talk and interact with other cells in the bloodstream. So we determine how much and how strong these markers are on your cell by flow cytometry. Then another value that we add on to your samples, we add the information from cytogenetics. And you heard this morning there's fluorescent in situ hybridization such as the 11q, 17p, trisomy 12, 11q, or 13q expression. not only do we know you have a 17p deletion, but we record in the database the percent of the cells. So out of 100 cells that we look under a microscope, we know 50% of those have the 17p deletion. Or later on in the disease, we know 85% of the cells have the 17p, or they do not, or have these other markers. And all of this information is recorded in the biorepository in the database, and it's given out to the scientists that ask, okay, Laura, we want 100 CLL samples with everybody with 17p deletion greater than 50%. And we want, like with those samples, we want to know their SAT70 data, VH status, immunophenotyping data. So I put all that together, all that information, and give it to the scientists for research. And of course, all of that could not happen as another level. We have the clinical data, and the clinical data comes from your disease course. I work closely with the data managers from all the CRC sites that Dr. Kipps described this morning, and they give me the follow-up data from when was the patient treated, when was the disease started, when was the minimal residual disease occurred. All that information gets added on to your samples, to the database, to this pyramid. Now, like I said, then the last step that happens towards the end of the pyramid, I distribute the samples to the scientists in the CRC Consortium. But I don't just distribute them whenever I want. We have a specific process. And I work very close with Donna Neuberg and Dr. Kipps to decide how many of the samples we can give out, what the inventory of the vials, and whether the project that the CRC investigator has requested is worthwhile. We call them hypothesis-driven studies. We want to make sure that the CRC investigator is asking to do a very specific study on those samples, because they're very valuable and I really take care of them very well. And I want to make sure that the information that the scientist gets will eventually lead to a publication, which is really the top of the pyramid. And you can see that the final purpose of why we do what we do is to define discoveries and publish papers that can help the CLL community, the world, and every other newly diagnosed patient to help cure this disease. So today in the TissueCore, we've been tracking almost more than 6,000 CLL patients, and we do the same profile here as you see with every one of the samples at several time points at diagnosis or when the samples come. Now, another power of the TissueCore and of this whole process is that we can follow these samples through time. As you give samples to us at the beginning of diagnosis, 6 months later, a year later, 2 years later, when you come into the clinic or when it's requested for clinical trials, we can do studies and we can look at the white blood count or these different aspects, and we can see how they change through time. Because we want to know, when does the kinetics of your disease change? And some people, as you can see, have a kind of an indolent disease. They have a type at the beginning, or during the disease course, they have the A type, where the white blood count kind of hovers, and as we keep getting samples throughout the years, they don't really go up very much. Some other patient samples have a different kinetics. For example, section B, you can see that it's very fast, rapid growth in the white blood count. And we look at that from all these markers that I described and the clinical information, and we see that things change very rapidly. So we have samples from time point 1, time point 2, and we can see, well, what happened in the genetics, the markers, the cytogenetics, what happened between time point 1 and time 2 in your cell? What made that change? But the very interesting cases are type C, because those individuals at the beginning had an indolent disease, and then all of a sudden things shoot up and things change at time point 3 or just before. So we have samples in the TissueCore at time point 1, time point 2, and time point 3. So the investigators can say, Laura, I want patients of type C kinetics, because I can look, well, they were indolent and something happened around time point 2 that changed their disease course. A novel mutation occurred, a cytogenetics abnormality occurred. I don't know. The scientist goes and figures this out. So we can then predict by the changes in the phenotype and genotype of these patients what changes the disease kinetics, and the doctors can, of course, give appropriate therapies or develop novel therapies or no therapies at all for patient A versus patient B or patient C. So this is the merit of continuing to collect samples in the TissueCore during the disease course of the patient. Now, I don't have many slides, but I always look at the TissueCore as a wine cellar for them. It's just to give you like a very basic analogy, because it's like it gets better with age. And I'm not a very expert in wine, but you can understand that a patient sample that has been in a TissueCore with serial samples for 10, 20, 15 years, that valuable sample is very valuable because it has a lot of annotated data, like I showed you in the pyramid, associated with that individual lifetime during the disease course, and then it'll help us all make discoveries much better. So I really have to thank all the patients in the CLL community for contributing your blood samples, and I assure you I take care of them very well today through Donna and Dr. Gibbs, and if you have any questions, you're welcome to speak to me anytime, and I can answer very specific questions. I have to thank the CRC members because, of course, like Dr. Gibbs described, we don't only get samples from UCSD, we get them from 6 institutions across the country. So this is a national bank, and it's a very national, very precious resource in the whole United States for CLL patients, not just in California, but all over the country in the CRC. And of course, I have to thank Dr. Kipp's lab that's involved, and you will hear later, in many of the discoveries of those samples that they take for hypothesis-driven studies. So now I will pass on to Donna, that works close with me, and she will tell you.
So the first thing I want to say, in answer to Tom's question this morning while I was sitting in the other room, yes, indeed, that is a magic 8-ball that sits on it. But I want you to understand that it has a very well-defined and known probability mechanism associated. Okay, so I thought of lots of titles for this, but really what I'm going to talk about is why do we so much want your blood, and why do we want so much of it, and why is giving it to us only once not enough? So we have to think of CLL as a unique disorder, the tumors in the blood, and that sample that you give us contains large numbers of tumor cells. That means that we can really not only look at that snapshot of your tumor at your first sampling, but if you'll come back and give us more tumor, we can look at it and see how it changes over time. It is one of the few diseases where serial sampling of tumor is feasible. And that puts us in a very special category, because when you talk to people who study solid tumors, they say, oh, you're spoiled. You work in CLL. You can actually look at the disease. Indeed, we are. And so what I decided to focus my talk on today was the question, have we really learned a lot about the disease from these efforts where people put blood into a bank, maybe our bank, maybe another bank, and have we really learned a lot about your disease, and does it have implications? And I think the answer to that is definitely yes. So I'm going to just give you a very brief retrospective over studies that relied on stored samples, finally on serial samples. And on the very first one, I'm not going to restrict this to CRC studies, but you all know about the FISH hierarchy of classification. It's associated with overall survival. And this is a figure from Hartmut Donner's initial paper 15 years ago in the New England Journal of Medicine. How could he do this study? He was at the University of Ulm in southern Germany, and someone had the forethought in the mid-90s to start storing samples from CLL patients. Not all taken at diagnosis, some obtained after they'd been treated, but those 325 patients who gave their samples resulted in this hierarchical classification, and everyone revisits this and says, well, does it still hold today in the modern treatment era, or the postmodern? I suppose we're entering postmodern. And the conclusion is yes. So that's a nice example of how much you got out of, I think that was a very limited amount of stored blood. Now, this is something that we actually did in the CRC. When we first started assembling data in the CRC and samples, we realized that we had some interesting ideas, and Laura is the first author on this. Tom's idea, but this is our ZAP70 paper. And we looked at IGHV mutation status in ZAP70 and found out that ZAP70 was in fact a predictor and associated with better outcomes for patients. How could we do this? We had a huge reservoir of samples that could be run, could be studied, and what you see on the bottom panel there are Laura's investigation of how her assay performed over time and also how constant ZAP70 levels were over time. So had we not had serial samples, we couldn't have seen that we were looking at a marker that seemed to really be where it was, with very little variability in patients over time. That's also a New England Journal paper. I think I sort of stuck with well, not quite. Now, one of our major users of the TissueCore has been Carlo Croce and his colleagues, and this is his initial paper on microRNA signature that's associated with prognosis and progression in CLL. This is by George Kalin, who was a student, a postdoc at that time. He's now a faculty member at MD Anderson. Carlo Croce was the senior author on the paper. But again, it's taking CLL and the understandings of CLL into the new world of microRNA. I'm embarrassed, but I'm going to use one of my own papers, and this is from the resource we had put together at the Dana-Farber. Kathy Wu and her postdoc, Mimi Wong, did whole exome sequencing. So now we're moving into the modern era of sequencing those samples. And in our first effort, we found genes that were mutated in CLL that were associated with differential clinical outcomes. But no one had reported those genes to be mutated in CLL. And the second from the top is SF3B1. It's a splicing factor gene. It had only recently been reported in myelodysplastic syndrome at the same time that we were reporting it in CLL. And finally, I'd like you to focus on the very colorful picture in the upper right corner. This is a paper by Anna Hsu. It's from the UK, and she had access to serial samples from CLL patients. The patient at the very top has 1, 2, 3, 4, 5, 6 serial samples, and what you can see if you just, well, for me, I just take off my glasses, but if you squint just a little, you can see that where the bar graph is very short, the third time point, that's after treatment. There's not much disease there. It's very hard to say what colors there are, but if you look before treatment, there are a lot of those red mutations. These are all the different mutations she found based on whole genome sequencing in the CLL. And what happened is that after treatment, I'm seeing a much bluer and more purple graph. That tells us that the mutational composition of that patient's disease is changing. Not that the blue and purple aren't there in the beginning, they are, they're the very tiny bands at the bottom. But the treatment seemed to have really been effective against the red mutations that are colored red, leaving space for those blue mutations to just mushroom after treatment. It's important that we know that. Now, the folks that I've worked with for a long time know that I would not omit an opportunity to teach you something about biostatistics. So I want to tell you what we learned from Anna Hsu's work and from other people who were starting to look at serial samples. The first plots that I showed you all start at time of diagnosis. But it is very rare for us to have your diagnostic samples in our bank. We usually get them when you come to visit someone who's part of the CRC, but you've been diagnosed in your own community. What we learned is that we really can't make inference. We can't understand what a patient's disease looked like out here at diagnosis if our first sample is over here. You come to us often when you need to see the specialist. Now, Brian would say that's as early as possible, but for some people it hasn't been that early. And so what we now do is we don't look at the implications for prognosis or progression of disease from the time of diagnosis. we look from the time of the snapshot of the sample that we were able to examine. And I think that's a paradigm shift in how statisticians think of disease, and probably CLL is the only disease in which we can implement that paradigm shift. So on behalf of the CLL researchers in the CRC and those of our colleagues across the world, we want to thank you for participating in clinical trials and for being patient with us when we again and again ask for samples of your disease. I think it helps us understand the disease better, and I think it's the key to a cure. Thank you.